The clinical landscape for COVID-19 treatment is constantly evolving. This video will walk viewers through the major therapies under investigation, highlighting their mechanisms of action and key studies so far. Currently, the Infectious Disease Society of America does not recommend that any of these drugs be used without a clinical trial. The information discussed here is up to date as of May 4, 2020. By the end of this concept video, healthcare professionals will be able to recall the basic pathogenesis and host immune response of a COVID-19 infection, describe the evidence basis for investigational therapies targeting the SARS-CoV-2 replication cycle, and the host immune response. This video is geared toward healthcare professionals already familiar with the basics of COVID-19. For a more extensive primer, please see curriculum.covidstudentresponse.org. The drugs discussed during this two-part video series are listed below with their trade names and relevant timestamps. In Part 1 of the video series, antivirals will be discussed. In Part 2, medications that act on the immune response will be discussed. Several investigational therapies for COVID-19 inhibit different stages of the SARS-CoV-2 viral replication cycle. First, SARS-CoV-2's spike protein binds to the host cell's ACE2 receptor, undergoes a conformational change, and is cleaved by the host cell's TMPRSS2 serine protease. The virus can then enter the cell either through endocytic or non-endocytic pathways and release its single-stranded positive sense RNA genome into the cytosol. This RNA can be translated immediately into viral proteins by host cell machinery. Some of these viral proteins complex to form a viral RNA-dependent RNA polymerase to produce new viral RNA. Viral proteins and RNA are then assembled into a new virion in the Golgi and are released to infect other cells. Some of the most buzzed about and controversial repurposed therapies for COVID-19 are chloroquine and hydroxychloroquine. These drugs have previously been used in the treatment of malaria and various autoimmune diseases such as lupus and rheumatoid arthritis. Their mechanism of action against SARS-CoV-2 likely involves inhibition of viral fusion to the cell membrane through pH modulation and decreasing viral membrane receptor glycosylation. The antiviral properties of chloroquine have been well established. It had known in vitro activity against other coronaviruses, and activity against SARS-CoV-2 was established in vitro in March. Later in March, Gautret and colleagues published a non-randomized control trial on hydroxychloroquine and azithromycin compared to supportive care alone on 36 total patients, reporting a significant decrease in viral load by nasal swab. However, multiple concerns have been raised over the validity of this study. No clinical outcomes were reported, and 6 out of 26 hydroxychloroquine-treated patients who had poor outcomes, including ICU transfer and death, were excluded from data analysis post hoc. That, combined with the author's previous history of publishing fraudulent data, cast significant doubt over these results. The FDA issued an emergency use authorization for hydroxychloroquine on March 28th. On March 30th, Chen et al. Released a preprint describing their RCT of 62 patients in Wuhan with mild COVID-19, suggesting a one-day faster time to improvement with hydroxychloroquine compared to standard of care. Adverse events were not reported. More recently, on April 14th, Tang et al. Released a preprint on an open-label multi-center Chinese RCT of 150 hospitalized COVID-19 patients that found no significant difference in likelihood of viral RNA clearance and no significant difference in symptom resolution over 28 days. The researchers did find hydroxychloroquine-treated patients had a significantly lower CRP and an insignificant trend towards faster resolution of lymphopenia, though these lab findings didn't translate to clinically meaningful results. Hydroxychloroquine-treated patients also had a higher adverse event rate of 30% compared to 8% in the standard of care arm. Magagnoli et al.'s retrospective preprint of 368 COVID-19 positive hospitalized veterans in the United States reiterated these concerns, finding that there was no reduction in need for ventilation, but actually an increase in overall mortality for those who received hydroxychloroquine compared to controls, though of course there may be selection bias in a non-randomized study. The American College of Cardiology and the FDA on April 24th cautioned clinicians to weigh hydroxychloroquine and chloroquine's drug toxicity in its use, particularly regarding QTc prolongation and risk of cardiomyopathy. Because hydroxychloroquine has a long half-life, risk of drug-drug interactions and side effects linger long after the last dose received. Dozens of other clinical trials are ongoing for hydroxychloroquine and chloroquine, including prophylaxis studies for those who have been exposed or are frontline workers. In an effort to consolidate research efforts, on March 18th, the WHO began its Solidarity RCT, which also includes Remdesivir and Lopinavir-Ritonavir plus/minus interferon beta. As of now, clinical data on hydroxychloroquine and chloroquine seems shaky at best, and some countries have discontinued its use for COVID-19 patients. In the US, clinicians should be cautious about weighing the possible risks and benefits of its use. Remdesivir is a nucleotide analog originally developed to treat Ebola virus and Marburg virus. It is owned by Gilead Sciences. It was found to have a favorable safety profile in Ebola virus clinical trials, but because it was less effective than monoclonal antibody treatments, it was ultimately not approved for this use. Remdesivir is an adenosine analog prodrug. The active compound is used by RNA-dependent RNA polymerase and inserts into viral RNA chains, causing delayed premature termination. Remdesivir was found to inhibit SARS-CoV-2 in vitro and provide clinical benefit in SARS-CoV-2 infected macaques. Grain et al. Studied remdesivir use in hospitalized patients with severe disease, though they excluded patients with severe renal or hepatic injury and did not have a control group. They showed clinical improvement in 36 of 53 patients and 7 deaths, with 18% mortality in their included ventilated patients compared to another study's statistic of 66% mortality in all ventilated patients. Interpretation of this data is challenging in the absence of a direct comparator group. A study from China, Wang et al. In Lancet, however, showed no significant clinical benefits, but had a trend towards time to clinical improvement. Unfortunately, this study was under-enrolled and underpowered because of decreasing COVID-19 cases in China. A press report later stated that an RCT conducted by the National Institutes of Allergy and Infectious Disease showed that remdesivir decreased time to recovery in patients with severe COVID versus placebo to 11 days compared to 15 days. Although the endpoint of the study was revised after the study started. Remdesivir received an FDA EUA on May 1, 2020. Remdesivir is one of the arms of the WHO Solidarity Trial. There are currently at least 7 clinical trials planned or underway with results expected later in May. As of now, clinical data on remdesivir in the treatment of COVID-19 is mixed, with negative data from the one published RCT and a press release citing positive data from another. However, there is much excitement about its use due to its activity in preliminary studies and favorable safety profile, and the WHO's R&D blueprint reports considers remdesivir the most promising drug candidate for SARS-CoV-2 for these reasons. Favipiravir is an RNA polymerase inhibitor approved in some Asian countries, including Japan and China, for the treatment of influenza. It is contraindicated in pregnancy due to risk of fetal harm. Favipiravir is a prodrug. Its active metabolite inhibits viral RNA-dependent RNA polymerase. In March 2020, an open-label non-randomized before-after control trial comparing Favipiravir to Lopinavir-Ritonavir in 80 patients showed that viral clearance occurred with a median of 4 days compared to 11 days in patients on Lopinavir-Ritonavir, and that 91% of people on Favipiravir had improved CT scans with few side effects. An ongoing study in China is comparing Favipiravir in combination with tocilizumab. A Favipiravir trial will be starting soon in 3 Massachusetts hospitals. As of now, clinical data on Favipiravir in the treatment of COVID-19 is sparse, with 1 positive non-randomized trial compared to Lopinavir-Ritonavir and no data from randomized controlled trials. Its teratogenic activity is a potential limitation, but there's excitement about its use due to these preliminary studies. Lopinavir and ritonavir, co-formulated under the brand name Kaletra, among others, are medications currently approved to treat HIV/AIDS. Both lopinavir and ritonavir are protease inhibitors, which inhibit the action of HIV protease. Although the SARS-CoV-2 protease Mpro is not structurally similar to HIV protease, in vitro studies showed lopinavir was able to inhibit Mpro as well. Ritonavir then works by slowing down the breakdown of lopinavir by the cytochrome P450 system in the liver. In March 2020, a randomized control open-label trial in 199 patients in Wuhan, China showed no benefit of Lopinavir-Ritonavir over standard of care in terms of viral load, time to clinical improvement, or mortality. However, a subgroup analysis showed a trend to improvement in patients who were treated earlier in their disease course with the drugs. Multiple trials are ongoing with Lopinavir-Ritonavir in comparison or in combination with many other drugs. Lopinavir-Ritonavir is involved in 2 of the arms of the WHO Solidarity Trial. One randomized control trial showed no efficacy for Lopinavir-Ritonavir, which makes its outlook less promising compared to other medications. However, it is still being studied earlier in the disease course and in combination with other medications. Known drug-drug interactions with ritonavir make administration of Lopinavir-Ritonavir potentially more complex than the other antivirals discussed. Finally, Camostat mesylate is a serine protease inhibitor used in the treatment of chronic pancreatitis, reflux esophagitis, and fibrotic liver and kidney diseases. As SARS-CoV-2 viral entry is dependent on activation by the host cell's serine protease, TMPRSS2, Camostat mesylate, as a serine protease inhibitor, would prevent viral fusion to the host membrane. Studies have confirmed this mechanism of action and that Camostat mesylate decreases SARS-CoV-2 infectivity in vitro. Multiple clinical trials are ongoing with no results published as of yet. There's not enough data to determine the outlook for this therapeutic, though its mechanism of action is theoretically promising. This video described the current evidence regarding 5 antiviral medications for COVID-19. Here's a graphic depicting the SARS-CoV-2 viral replication cycle with the targets of the drugs we just described. We have also compiled this information into a table for your convenience. You can download this at the link below. Thank you for watching.